Hi, and welcome to this lecture on viruses. A brief word about how to go through this video. This video uses PowerPoint slides, but it is not a chalk and talk lecture. There will be moments of pause where you are directed to answer a question, draw something, or otherwise participate. I do not have many words in my slides, and the few that are there are to help you if you need to go back and review. Um, As you play the video, try to look at the pictures and listen to the sound of my voice. Take notes as you need to. Do not try to read the slides while you are listening to my voice. If you want to read a slide, pause the video and then read the slide. However, I do encourage you to practice active listening as you watch the pictures. If there are important words on the slides, I will say them out loud or point you to them with time to read. Okay? Give it a try. Viruses are acellular, meaning they are not made of cells, and infectious. They are obligate intracellular parasites with host and cell type specificity. This means they can only reproduce inside another living cell. Although all viruses are infectious, not all are pathogenic or disease-causing. Viruses were not seen until the late 1930s when the electron microscope was invented. Prior to that, the existence of viruses was only confirmed by transmission. This means that if doctors or scientists could not find evidence of bacteria causing an illness, they would take a blood or fluid sample from a patient and inoculate a test subject, usually rabbits, mice, hamsters, or other small laboratory animals, and then see if those animals also develop symptoms or die. Viruses consist of genetic material in the form of a nucleic acid molecule, either DNA or RNA, never both, usually in a protein coat, and viruses are capable of directing their own reproduction and spread by co-opting the machinery inside the host cell it invades. The ability to analyze DNA and RNA of viruses gives us a complex picture of the virus genetics, and this can be used to classify viruses. So for taxonomy purposes, viruses have genus and species names. Now let's look at the characteristics of life that you may remember from GenBio. For living organisms, they must be organized, they must respond to stimuli, they must reproduce, grow, and develop. They should be able to adapt to their environment and process energy on their own, and they can maintain homeostasis. So these are the characteristics of living organisms. Viruses cannot reproduce, nor can they process energy. They have to take over the host cell's machinery to do that. And although they can adapt or mutate to increase their chances of replicating, they cannot grow or develop. So since viruses do not fulfill all the requirements of living things, we can see that they are not living things. One thing all viruses have in common is that their genome is surrounded by a protein capsid. Many even have an additional phospholipid membrane studded with glycoproteins as seen in this figure. Here we see common virions. This is the word we use for virus structures. The first three are what I guess we'd call typical virus structures. The one on the far right is a bacteriophage. Bacteriophages are viruses that only infect bacteria. Viruses can infect every type of host cell. This means plants, animals, and even prokaryotes. Viruses that infect bacteria, again, are called bacteriophages, and we'll talk about this a lot later. But phage comes from the Greek word for devour, which I think is kind of cool. Generally, viruses have one mode of infection for a particular cell. For instance, the tobacco mosaic virus, seen here in figure A, only infects tobacco plants. Humans nor animals can contract TMV because they do not have tobacco leaf cells in their bodies. However, as we've seen recently with COVID-19 jumping from bats to humans, sometimes viruses can be transmitted from animal host to human host. These are called zoonotic viruses. Zoonosis is very rare, but as we are seeing now with COVID and previously with swine flu and mad cow, it can happen. In reverse zoonosis, human viruses become infectious to animals. We have seen a few cases of this with COVID-19 infections in tigers at the Bronx Zoo and some house cats and recently one pet dog testing positive for COVID. One of the ways viruses are categorized is by general composition. Figure A shows a naked or non-enveloped virus. It has nucleic acids, in this case DNA, inside a capsid. The capsid is spiked with glycoproteins. Altogether, it's a very simple structure. Figure B shows an enveloped virus. In addition to the nucleic acid-filled capsid, in this case the nucleic acid is RNA, it also contains a viral envelope in addition to the glycoprotein spikes. These viral envelopes are typically made from the nuclear envelope or cell membrane of the host cell. So just imagine if your bed is the host cell nucleus and the sheets are the nuclear membrane and you are lying under the sheets. You are the virus. When you, the virus, is ready to emerge from the cell, it covers itself with a sheet and leaves the bed. The nucleus bed has now lost its viral membrane and must die, and the virus has a new envelope and can move on to infect another cell. This is how white blood cell counts get so low in HIV. The virus kills the cells by stealing pieces of the nuclear membrane when it emerges after taking over the nucleus to replicate, and this causes the white blood cell to die. Another way to categorize viruses is by their shape. Viruses can be either helical with a cylindrical capsid, like figure A, polyhedral or many-sided, like figure B, or complex, which really means everything that's not distinctly helical or polyhedral, like figure C. Helical and polyhedral viruses have capsids and maybe an envelope. Complex viruses may not have a capsid or an envelope, like bacteriophages, or they may have both a capsid and an envelope, but no organized outward shape. Pause the video here and sketch these three types of viruses on a sheet of paper so that you may refer to them when we discuss viral life cycles. Put these in your notes. All viruses depend on cells for reproduction and metabolic processes. They don't have the necessary enzymes or proteins for replication. This is why they need to take over the host cell reproduction apparatus to do this. The whole purpose of a virus is to make more virus. That's it. Viral replication occurs in the cytoplasm in bacteria and in the nucleus for animal cells, with a few exceptions, like large viruses that may be bigger than the nucleus or RNA viruses. Virulent phages kill the cell by lysis, which means bursting the cell. Temperate phages take over the cell machinery until they are ready to make newly assembled viruses, kind of like zombies. In the case of temperate phages, the virus combines with the host DNA or RNA and becomes part of the cell's chromosome. Each time the cell replicates, it now replicates the virus genome. Then, when the virus is induced, the viral genome breaks away from the host chromosome and exits the cell, generally resulting in cell death. So imagine if one infected cell replicates 100 times, then you have that many completely assembled progeny viruses released all at once, which will lead to massive host cell death. First, we will discuss the life cycle of viruses with prokaryotic hosts. Then we will discuss viruses with eukaryotic hosts. Prokaryotes have 3 viral life cycles. We'll start with the lytic cycle. In the lytic cycle, a virulent bacteriophage takes over the cell, reproduces the phages, and destroys the cell. Here we see the 5 stages of the lytic cycle. In the first phase, attachment, the virus uses the tail fibers In the lytic cycle, a virulent bacteriophage takes over the cell, reproduces more phages, and destroys the cell. Here we see the 5 stages of the lytic cycle. In the first phase, attachment, the bacteriophage uses the tail fibers to attach to specific bacterial surface receptors on the host cell. During the penetration phase, the tail sheath pokes through the host cell wall and cell membrane like a hypodermic needle and injects its genome into the cell. During biosynthesis, the phage disintegrates any part of the bacterial chromosome that it doesn't need, leaving only the machinery necessary to replicate, transcribe, and translate new viral components. These components are assembled to create new bacteriophages during the maturation phase. And finally, the cell lyses to release the progeny viruses into the environment so they can infect more cells. This kills the host cell. A temperate bacteriophage has both lytic and lysogenic cycles. In the lysogenic cycle, phage DNA is incorporated into the host genome, forming a prophage, which is passed on to subsequent generations of cells through cell replication. Environmental stressors, such as starvation or exposure to toxic chemicals, may cause the prophage to be excised and enter the lytic cycle. This activation of the lytic cycle is called induction. In this case, since the viral genome is already part of the host chromosome, the lytic cycle picks up at the maturation phase of the lytic cycle by assembling, then releasing new bacteriophages. Transduction occurs when a bacteriophage transfers bacterial DNA from one bacterium to another during sequential infection. There are two types of transduction: general transduction and specialized transduction. In general transduction, a random piece of the host chromosome is transferred with the phage during the lytic cycle, so a kind of hybrid chromosome is created. In specialized transduction, the prophage and a specific piece of the host cell's genome is transferred during the lytic cycle. This flowchart illustrates the mechanisms of transduction. In transduction, a bacteriophage basically follows the lysogenic cycle. Then, instead of releasing new phages during the lytic cycle, the phages released also contain pieces of the host cell's DNA. Eukaryotes also have a lytic cycle of viral replication that follows similar stages to bacteriophages. However, instead of piercing the membrane to deposit viral genetic material like a hypodermic needle, viruses that infect eukaryotes enter the cell by endocytosis or membrane fusion. Endocytosis is shown here in the figure. We see the virus lands on the host cell and then wraps itself in the host cell membrane as it barges inside. Kind of a reverse for our bedsheet metaphor. With membrane fusion, the virus lipid membrane would merge with the host cell membrane and then kind of pour its genetic material inside the host cell. Like bacteriophages, these viruses have super host virus specificity. We call this tissue tropism. For example, influenza virus is specific to or has tropism for the cells of the respiratory tract. HIV has a tropism for white blood cells. One thing that is fascinating about COVID-19 is that it doesn't seem to have a specific tropism. The more we learn about COVID-19 infection, the more tissues seem to be susceptible to infection. Anyway, back to the lytic cycle. I've mentioned that the mechanism of cell attachment is different than lytic cycle for bacteriophages. Replication is also different. If you look at step 3 here in the figure, you'll see that once the virus is inside the cell, the virus loses its coating, causing all of the contents of the virus to be released into the cytoplasm. From here, the viral DNA, or RNA, enters the nucleus for replication in stage 4 and then reassembles into new particles as seen in stage 5. Finally, once new phage particles are made, viruses are assembled and released by budding, which means that they pinch out a little bit of the host cell's membrane to make a new viral membrane on their way out of the cell. The cell is not killed during this process and continues living to produce more virus. So now we know a little bit about viruses that affect eukaryotic hosts. Humans are eukaryotes. This is a good time to talk a little bit about viral infection in humans or even other animal hosts. Infections can be acute or persistent. Acute infections cause illness as soon as a person becomes infected. Influenza virus is an example of an acute viral infection. People tend to get sick within a couple of days of exposure to an acute virus. There are two types of persistent infections, latent and chronic. Latent viruses do not go through the lytic cycle. This means they replicate and then remain hidden inside the cell until they are activated. Latent viruses may cause acute infections and then become dormant. So even though a person is no longer feeling sick, the virus is still active in their body, just hiding in their cells like a Trojan horse waiting for its moment of attack. Sometimes latent viruses infect cells and hide without causing initial symptoms at all, only to spring out and make you sick days or even weeks later. People with latent viruses can sometimes be contagious even though they're not showing symptoms. Herpes simplex virus, which causes oral and genital herpes, and the varicella zoster virus, which causes chickenpox and shingles, are examples of latent viruses. These are two viruses that we know don't cause symptoms at the moment of infection. They have an incubation period, and symptoms don't show up for several days, or even weeks in some cases. In the case of shingles, after a childhood bout of chickenpox, the virus reawakens after decades to cause shingles. People with latent viruses are usually unaware that they are carrying the virus unless they've been tested. Plant viruses are similar to the animal viruses that we just discussed. They are transmitted by contact between plants, fungi, and nematodes usually found in the soil. They can also be transmitted by insects or other arthropods and wounds, like from pruning or wind damage. Some plant viruses have a very specific host range. Some even have a specific vector host range. For instance, a particular virus might be transmitted only by ants to a certain tree, but not by aphids. Plant viruses are biotrophic parasites, which means they can infect without killing the host. Infection might occur immediately for lytic viruses or later for latent viruses. Finally, we'll talk about some not quite viruses that can also be infectious. Virioids are short strands of circular RNA that can self-replicate. Like viruses, they do need a host cell for replication machinery, but they do not have a protein coat. Virusoids are non-self-replicating particles. They require a secondary infection of a helper virus to help the virusoid take over the replication machinery. Prions are infectious protein particles. They are made from misfolded forms of normal proteins found in the cell. Prions are responsible for mad cow disease. They are extremely difficult to destroy because they are heat, chemical, and even radiation resistant. Now for your homework. Look at the three viral structures in the diagram. Use these virus structures as inspiration to design your own virus. Draw the virus, label the structures of the virus, and then describe the following. What is the mode of infection? What is the host cell type? And what are the symptoms if a person becomes infected? You may mix and match components from each of these viruses for your virus creation. You can also invent something new. It's up to you. Be creative. When you finish your design, upload it in the Assignments tab under the Design Your Own Virus assignment.